Hi everyone, my name is Ember Bonini, and today my group mates and I are going to be talking about hyperglycemia in stroke and how it impairs polarization of monocytes and macrophages to a protective inflammatory cell type. So right here we have who the authors are, and then these are the names of our group mates. Alright, so this is our day one information. So to start off, we're going to be going over some background knowledge. So the first thing you guys need to know is what hyperglycemia is. This is when an individual has high blood glucose concentration, and it can be caused by having an insane amount of insulin in the body or just not responding to insulin properly. For an individual who has hyperglycemia, they can either have pre-existing diabetes mellitus, or if they don't, it can be caused by stress. When the body is under stress, it causes a release of hormones like cortisol, glucagon, or growth hormone. These take the body out of homeostasis, causing the hyperglycemic conditions to occur. So as we look into the pictures right here, these circles, the little blue ones, that's just showing the amount of blood glucose that's supposed to be in your body, and then when it's low, there are fewer circles. As you can see, our normal, it has an even mix of them, and high right here, it just has an increased influx of the glucose and different blood sugars in there. Next. All right, the next thing to note is what an acute ischemic stroke is. So this is when there is a sudden loss of blood circulation to a specific area of the brain. This is not found in all areas of the brain at once, but more so just one area at a time. If you look at our picture, it's showing that in this area is where there's a loss of blood flow to it, which is why it has the darker coloring in it. So when you're looking at an acute ischemic stroke, it can be found through looking at a computed tomography or a CT scan, and this just helps to see if there's a brain clot or blockage of blood and where it's found in. Some of the things that are seen in individuals with ischemic stroke is a loss in neurological function. So common symptoms of this and what it can look like is having weakness in your limbs, vertigo, or balance issues, and you can also have issues with speaking or trying to understand different words. Then we go into the polarization of monocytes and macrophages. So it's important to note of what a monocyte is, and this is just a type of white blood cell, whereas a macrophage, they can modify their phenotype in response to a different stimuli. So if we look at the picture in this one, we can see it right here that a stimuli is being sent onto a monocyte, which is causing it to change into either a macrophage or dendritic cell. And we don't really need to focus on the dendritic cells right now because we're more so looking at how macrophages are being polarized and what's happening there. And a macrophage is used to detect and destruct different harmful organisms or different damaged cells within the body. And finally, we have the inflammatory process. So the inflammatory process, in a short summary, is how the body responds to injury or infection. So this can be seen as different chemicals are being released from damaged cells, causing the blood vessels to, in a way, leak fluid. As we can see in the image here, we have our capillary bed, and then within it, if we look at the second form of the picture, it has fluid, which is in the form of these like yellow globs, and that's being brought into the different tissues, which is causing that inflammation to occur. So then these are sending a signal to white blood cells, so that way they can commit phagocytosis and eat and mark the damaged cells. Within this, it's also showing how depending on the stimulus they receive is how they go through phagocytosis. And then if the inflammatory process is damaged, it can cause things like chronic inflammation, autoimmune diseases, tissue scarring, or even systemic effects. And then one thing to kind of put this into perspective is if you sprain your ankle and it starts flaring up and growing in size, that's just showing how the inflammatory process is occurring. And then as it goes down in that different inflammation, and as it's going less from that swelling position, it's just the inflammatory process is working properly.
All right, so I'm Gaby, and I'll be presenting the first observation that was made. And for the first observation number one, it's basically hyperglycemia is especially strong in non-diabetic patients. And as Ember described before in the background knowledge, is that basically hyperglycemia is, the rise of blood glucose levels, and it's mostly present in diabetic patients, but clinical evidence suggests that hyperglycemia can arise in non-diabetic patients in the form of acute hyperglycemia. And basically, what the difference between acute hyperglycemia and chronic hyperglycemia is basically, as it suggests, acute hyperglycemia is basically it's a short-term rise of blood glucose levels, which is mostly present in non-diabetic patients, which can arise from stress, poor eating, or like any other like process that like makes the blood go higher. And this suggests that acute hyperglycemia is a causative factor in ischemic brain damage. And this was proven by clinical studies on rodents that confirmed this. And from this, current guidelines from the American Heart Association recommended treating hyperglycemia patients with acute stroke.
Hi everyone, I'm Maya. Alright, so I'm going to be talking about observation 2. So another one of the previous research studies talked about this B-hydroxybutyrate, which is an alternate energy substrate that is seen in inflammatory responses. So this helped our researchers understand more of the inflammatory process, and it also helped them to find these inflammatory and non-inflammatory markers that can help to monitor the process of inflammation. And so in a normal process, these inflammatory markers are recruited to the site of stroke in the central nervous system, and they differentiate into non-inflammatory cells from the inflammatory cells in order to protect the brain from damage that happened during the stroke. So this gave our researchers the means to track the inflammatory response, and it also prompted them to find the relationship between the stroke and M2 type macrophages to glucose levels, and that's what they're going to be mainly monitoring in this study.
Alrighty, and now I'm going to be going into observation 3. So another previous research study showed that acute hyperglycemia can aggravate ischemic brain damage. So this was just showing that there hasn't really been a consensus on hyperglycemia being a cause or like the main cause for poor stroke outcomes, but it has shown a correlation to it. And this has been shown previously due to hyperglycemia putting more pressure on the blood and different blood vessels, causing an increase in friction, which can lead into brain damage due to the flow of the blood going into the brain. And with this, it's also related to stress. As an individual gets more stressed out, it is still increasing that impact on the blood vessels. And this also increases the availability of glutamate, and glutamate has a direct increased response to calcium-mediated neuronal cell death, which obviously you don't want cell death within the brain, as it can lead to different damaging functions and neurological damages to the body.
Hi, I'm Makana, and I'm going to be discussing observation number 4. So this observation revolves around hyperglycemia treatment for patients experiencing stroke. So the basis of this is our researchers were looking for how this was treated, and they saw that the American Heart Association and the European Stroke Organization recommend treatment for patients experiencing hyperglycemia with acute stroke. But with further research, despite using insulin, which is the usual treatment for diabetic patients, it was not showing improvement in the outcome of acute stroke patients. Rather, it was causing harm and putting them at risk for hypoglycemic episodes.
Hello everyone, my name is Brad. I'm going to be presenting observation number 5. So in this experiment, there were two main cell types that they grew. There's the primary brain endothelial cells and the bone marrow derived macrophages, or the PBECs and BMDMs respectively. So these primary brain endothelial cells were prepared from 8 to 10 week old mice that did not have the genetic identifier that we're looking for. And the bone marrow derived macrophages were also prepared from the same mice strains, but they were grown in 25 millimoles of glucose to simulate the hyperglycemia, and then switched to a lower level of glucose, and they were polarized into the inflammatory and non-inflammatory phenotypes that we're going to be looking at later, or the LPS and the IL4 respectively. This setup helps understand how hyperglycemia affects macrophage behavior and polarization in these stroke conditions. So for observation number 6, we're going to be looking at the beta dicarbonyl levels in plasma and brain tissue of mice. The plasma was collected before there was the MCAO, or the middle cerebral arterial occlusion, 30 minutes after and 50 minutes after as well. And the brain tissue was taken from the same brain before and harvested and snap frozen in liquid nitrogen. So these beta dicarbonyls levels are actually important because they're the precursors to the end products that cause the hyperglycemia induced damage, or the things that just make it worse for the stroke patients when they have hyperglycemia. But this technique allows researchers to track changes in the beta dicarbonyl levels over time and in different tissues, providing insights into the metabolic changes occurring during a hyperglycemic stroke.
So the overall hypothesis for this research is that acute hyperglycemia downregulates protective non-inflammatory monocytes and macrophages in ischemic brain tissue. As stated before, both macrophages and monocytes are responsible for protecting the brain after ischemic stroke, and furthermore, the researchers in this article provide evidence that alpha dicarbonyl glucose metabolites and receptor for advanced glycation end products are potential mediators of glucose on the effect of non-inflammatory macrophages, which can potentially suggest new treatment strategies for hyperglycemia and hyperglycemia strokes.
All right, guys, I'm back. I know you missed me. I'm here to talk about key terms. We've already gone over some of them. I just want to talk about them a little more in depth, especially relating specifically to our study. So, as Ember talked about earlier in the background, cerebral ischemia is talking about restricted blood flow and decreased oxygen to parts of the brain that lead to cell injury and death. And so common in ischemic stroke, which is what we're looking at, is due to blockage in brain arteries. And later I'll talk about how the mice undergo that stroke experience. And so then going into endothelial cells, these are the ones that are lining the blood vessels, and they control things like blood flow, vessel permeability, and immune trafficking. And in this study specifically, the endothelial cells are influencing the polarization of the macrophages, which we talked about a little bit already, but we'll go into more with the figures. And then inflammation, which Ember also talked about a little bit in the background, affects the cell permeability. And the significance of this in our study is that there's a threshold that inflammation can reach from being good and cleaning up the cell damage to being too much and causing permanent cell damage. And we'll go into that a little bit more with the hyperglycemic versus the normal glycemic. And then neuroprotection is how the brain is protecting itself from injury. And in this study, the researchers were aiming to understand how hyperglycemia interferes with this exchange. And under the observation of certain immune cells, which is the monocytes and the macrophages that we're specifically looking at. And the macrophages themselves are white blood cells, and what their physical job is engulfing and digesting cellular debris, foreign substances, and pathogens. So that's the whole good part of the inflammation system of that whole cleanup process. But they also can be harmful, and so there's kind of a balance between these two, which is what the researchers are specifically looking at. And finally, we've got the agar, and this is the specific gene that we're looking at. So this receptor is on immune cells, and they bind to these AGEs, which can be harmful, and they're seen in the hyperglycemic response. And so the researchers are hypothesizing that if there's not the presence of this gene, then there'll be better differentiation from the inflammatory to the non-inflammatory type cells to then help with this brain damage. All right, moving on. We've got our techniques. You thought you got rid of me? Not yet. I'm moving on to mice. Wow. This is our first technique. It's the main technique overall. You're going to be looking at the mice under experimentation with the middle cerebral artery occlusion, which I will talk about more with the stroke model. But first of all, the purpose of using mice is to have a live model to be able to observe these biological processes, specifically with the inflammation. And so first of all, in the process, we wanted to separate these into groups, or the researchers did. And so this meant age and sex matching them, as well as doing wild type and non-wild type, and that's what this here, let me draw on it real quick. That's what this here is talking about, is the non-wild type, which you will see later on in figures as well, and I'll continue to point that out. But the researchers were blind to the genotypes, which is also seen in like placebo studies to help reduce bias when it comes to these research processes. And the mice were also put into groups of hyperglycemic versus normal glycemic to study these glucose levels under different inflammation. And the glucose was induced through an injection that happened right before the inducing stroke process, which I'll also point out later in one of the figures. And then they used genetically modified mice lacking that gene that we talked about earlier. Again, hoping that these ones that were lacking the gene would show more polarization from the inflammatory type to the non-inflammatory type. And these mice lacking the auger are also the wild type mice as well. So moving on to specifically the stroke model, the purpose of this was to induce stroke in the mice itself. And Brad talked about this a little bit earlier with the MCAO, which stands for the middle cerebral arterial occlusion. So this process specifically used what you can see here. This picture is a little different than what they did, but I just wanted to make sure you guys could see where the middle cerebral artery is in the mouse. And so first they were put to sleep with pentobarbital, which that's significant because the pentobarbital does not influence glucose levels, and because they were influencing the glucose levels prior to this procedure, so they didn't want anything to happen later on to then influence the normal glycemic mice to be more hyperglycemic. They wanted to keep them controlled. So basically they used this microbipolar electrocoagulation, which is kind of a fancy way to say cauterization, if you know what cauterization is. And so right here I put this little pair of tweezers to represent this. And so basically this is coming into this middle cerebral artery right here, and they would pretty much like snip it so that it couldn't have blood flow through it, but it wasn't completely cutting it so that the mice were like hemorrhaging. And then once this happened, the mice were monitored for tissue samples after 48 hours. I'll also talk about more of that experimental process once we get into the figures as well.
Hi guys, I'm back. So now I'm going to be talking to you guys about immunofluorescence microscopy, which is our third technique or assay that we're going to be addressing. So the purpose of this one is to visualize the location and distribution of proteins within a cell. And for this one, it goes over into a 2-day process. So I'm breaking up the procedures day by day. So on the first slide, we're just going to go over day 1. So for this first day, we're looking at ARG1, which stands for arginase 1 staining, and we're going to go over that into a figure in a couple slides later on. Alright, so within the arginase 1 staining, we're going to be looking at agar1, or it can also be represented as CX3CR1GFP, and just to kind of highlight that on the slide, it's right here is what we're talking about. And then it's looking into the WT mice, and that WT, which is right there, that just stands for wild type. And so the first step of this is putting these wild type mice into anesthesia through tribromoethanol. And then from here, they're going to be perfused with Ringer solution and then 4% paraformaldehyde. And then that's going to be abbreviated as PFA. So later on when you see it, that's just kind of what it's referring to. And then after we go through this arginase 1 staining, we're going to go into coronal cryosections. And for this, it's just looking at them being air dried for an hour and then rehydrated in another form of solution that's just known as PBS. And they're going to go through this for 5 minutes. And then overnight incubation is going to occur for these cryosections, and that'll be at room temperature. And then they use a couple different cells in like different, and they just use different solutions, and that can be seen through this GoTo anti-mouse arginase 1, and then they also use rabbit anti-mouse, and that's just known as IBAL, which is right there. And then really quick, we're just going to look at this image. So as we can see, we have a couple different sections. We have like this red coloring, but then we also have green. We're going to see a lot of this green, and it's just going to be known as and that is known as green fluorescent protein, and you're going to see that a lot come into the later figures. Now on to day 2 of this. So the next part we go into is incubation, and then the image that we have on this slide, it's really just showing the different microscope that they use. And so you put your slides into the microscope, you're going to zoom in on it, and then it's going to read onto this computer where you can see the different stainings on here. As you can see, our green staining is right in these corners on this image, but every staining you do, it is going to have like a different stain depending on what you're looking for. Anyways though, so day 2 is when our slides were undergoing incubation. So in here we have our cyanase 3 labeled anti-go, we have Alexaflor 488 labeled anti-rabbit, 647 is another form of anti-goat, this one is just using that Alexaflor, we have 594 anti-rabbit, and then we also have CY2, which is anti-chicken antibodies, and then these are just different forms that the slides are going, so that way they can test and see what the different results and reactions are going to be to those wild type mice. And then we also have some imaging on a confocal microscope, which is what this microscope is. That's the scientific term for it. And within this microscope, it's just going to count the immunostained cells on there, and it'll break down the different sections of them. And then it's going to show into predefined fields, and these different fields, which are also seen in the coronal cryosectioning, is we have our motor cortex, our striatum, and then our insular cortex. Alrighty, so our next technique we're going to talk about is bone marrow transplantation. And this one, it is pretty straightforward. What the title of this technique says is realistically what's going on. So the purpose of this technique is to just replace that damaged bone marrow with healthy stem cells. And during our research, it's looking at this procedure going in through mice, as that was our case study type. We were not performing this on humans yet. So the procedures for this one is the mice are being anesthetized with isoflurane, and then from there they're going to be decapitated, which is why we don't do this on humans. From here, we're going to have the femurs and tibias being aseptically dissected, and then the bone marrow is going to be flushed out. And again, this is also going to be dissolved in the PBS solution, which is what we saw in that immunofluorescence microscopy as well. And they're going to use a 23 gauge cannula. And then if we look at the image right here, it's just kind of referring to where the bone marrow is located in the bones. So you can kind of use that to just kind of imagine where it is on a mice and how they have to, how big that 23 gauge actually is to get into the bone marrow right there. From here, we're going to look at homogeneous cell suspension, and that is going through a 40 cell strainer. After this, our cells are going to then be centrifuged and suspended again in that PBS solution. Why they're using the PBS solution for both types of solution that they're going through is just to keep it as simple and constant as they can. So that way there aren't any errors or any different factors going into it. And then from here, they're going to be retroorbitally at an 8-week-old. And then again, we see our C57BL6 mice. And then those are lethally irradiated 1 day before they go through this retroorbitally. Finally, from this one, we have 6 weeks after that transplant. That's when they're going to look through the MCAO and kind of perform that. And they're going to go with this under normal glycemic and hyperglycemic conditions. This will be explained a little bit more into our figures, as this is the main study group we're looking at. We are looking at the normal glycemic and hyperglycemic mice.
I'm going to be discussing the technique of flow cytometry. So the overall purpose of this process is that it allows researchers to assess the immune cell response in the brain following a stroke, which is crucial for understanding the effects of hyperglycemia and other factors on stroke recovery and inflammation. So the first step is going to be blood sample collection. They took 100 microliter blood samples either before or 48 hours after the the MCAO process. Next, the mice were anesthetized with tribroethanol, which does not interfere with the glycemic levels of the blood. And 48 hours after the MCAO, their hearts were perfused with a Ringer solution, and this just clears blood from their brains. After that, their brains were dissected and the olfactory bulbs and cerebella were removed. The left hemisphere was then processed for cell extraction. So the brain tissue is digested in a medium called Dulbecco's modified eagle medium, or DMEM, and this contains collagenase and DNase. This was done for 30 minutes at 37 degrees Celsius, and this just breaks down the tissue and releases the cells for the analysis process. So next up, you're going to be filtering out the blood. So they used a 40 micrometer nylon cell strainer, and this is just to remove the larger pieces of debris. After that, the remaining blood cells are going to be lysed on ice using erythrocyte lysis buffer, and this is going to be used to isolate the immune cells. After that, a Perkel gradient was used, and this just separates the cells from myelin and other debris, and it allows researchers to collect the desired cell populations from the gradient interface. After this, the cells are going to be washed. So they're washed with 10 microliters of PBS, and this is going to contain a 0.5% BSA, and it's treated with a blocking reagent of rat anti-mouse CD1632. This is going to be done for 10 minutes on ice, and it's just put on ice to prevent non-specific binding of antibodies. Next up is going to be antibody staining. So the cells are going to be incubated with antibodies of their respective isotope controls. This is going to be done for 45 minutes still on ice. And the purpose of this is to identify different immune cell markers. So as we have listed here, we have CDC45, LY6C, CD11B, and LY6G. These markers are identified immune cell markers, and it just helps distinguish various cell types involved in immune response. So the last step of this is going to be the stain cells are going to be analyzed using a flow cytometer Pertec Cube 8, and this is going to be using a laser, kind of like how it's shown right here. So the purpose of this laser, it uses lines from 488 and 638 nanometers, and they're used to detect fluorescent signals from the antibodies, which gives data on the different population of immune cells present. So as you can see, the laser goes through, and here's all the little markers, and that is how it's going to be analyzed.
So with the preparation of the BMDMs, it just includes pulling that bone marrow out from femurs and tibias of the mice, and then they flush the bone marrow with sterile PBS or phosphate buffered saline. After that, they take that solution and they pass it through a 70 micrometer cell strainer that just removes the bone fragments and the larger cell clumps. And then they go back in and they remove the red blood cells using ammonium chloride, which just lyses them and leaves them out so that it won't be affecting the culture of the rest of the cells. And then they culture the BMDMs in a very specific solution in specific conditions with 25 millimoles of glucose, which is like a hyperglycemic effect, and then 10% FCS, which just provides growth factors for the macrophages to differentiate, and then antibiotics, which prevent contamination so that no diseases or bacteria actually grow in the sample, and then they treat it with CSF1, which stimulates that macrophage differentiation that will eventually lead to them being the BMDMs, or the bone marrow derived macrophages. And then on day 3, after they've been treating that for a while, they switch from the 25 millimoles to 5 millimoles of glucose, which is a normal level for the mice. And then on day 7, they stop treating with the CSF1 treatment and polarize the macrophages fully in that time. And then for the PBECs, or the peripheral, primary brain endothelial cells, were challenged with the methylglyoxal, or MG, and subjected to oxygen glucose deprivation, or that OGD that you're going to be seeing through the rest of the presentation. This mimics the conditions of an ischemic stroke with hyperglycemia, and the resulting condition medium was used to treat those BMDMs that we're going to talk about more later. This helps us understand how the endothelial cells influence the macrophages during an ischemic stroke.
Yeah, so I did not mean to sneeze or anything.
So this slide, we're going to be looking at the measurement of the alpha dicarbonyls. So this is just using that liquid chromatography and mass spectrometry that I had mentioned earlier. But instead of looking at alpha or beta dicarbonyls, it's just alpha dicarbonyls, which is realistically no difference except the location of the carbons in the stroke model. But it's the same process where it's collected, plasma's collected before the stroke, 30 minutes after the stroke, and 50 minutes after the stroke. And then the brain tissue from both hemispheres is sampled. They're homogenized in cold PBS, which is that phosphate buffered saline that I just mentioned. And that PBS is just a standard buffer solution that just keeps it a constant pH, so nothing, is too acidic for the cells to be looked at. It just makes it suitable for various biological and biochemical procedures. And then the protein concentrations were determined using Lowry's method. But all samples were derivatized from 1,2-diaminobenzene to stabilize the alpha dicarbonyls, and we used a reversed phase C18 column with a water acetonitride gradient containing 0.1% formic acid. The mass spectrometer used electrospray ionization in positive mode with detection by multiple reaction monitoring for high specificity and sensitivity. This method allowed us to quantify the beta dicarbonyl glucose metabolites, which are those precursors that I had mentioned earlier during the observations, and these metabolites were found to be elevated in hyperglycemic conditions and provided crucial data for understanding the mechanism by which hyperglycemia worsens stroke outcomes.
Okay, so the next technique is OGD and CSF-1 ELISA, and basically the overall purpose of this is to investigate the effects of OGD on PBEDs and assess the production and secretion of colony-stimulating factors in response. And those colony-stimulating factors are going to be explained throughout the presentation, but for now, for the procedures for this technique, basically primary brain endothelial cells, or PBECs, and they expose them to oxygen glucose deprivation, or OGD. The medium is changed to a glucose-free DMEM containing EC growth supplement and 2-deoxy-D-glucose at 5 at 5 millimolar, and the cells were treated with MG at 500 micromolar and for 1 hour, and transferred and sealed these cells into an anaerobic chamber containing 5% of CO2 and 95% of nitrogen for 15 minutes, and then they incubated those cells for 4.5 hours at 37 degrees Celsius. Then they switched back those cells to EC medium for 4 more hours before being lysed for mRNA preparation. And just a quick definition for those, so then you know what lyse is. It's basically the undergo breakdown of the cell, and they're going to use that for mRNA preparation. Then they collect that medium for BMDM treatment or CSF-1 measurement by ELISA. And for those of you who took lab probably already know what ELISA is, but just in case, it's basically a test that detects antibodies, antigen, proteins, etc. And for this is basically, we're going to use it to measure CSF-1. And
now we're going to be looking at real-time RT-PCR. So the purpose of this is to isolate RNA from specific cell types and using it for quantitative reverse transcription PCR. This is also going to be called QRT-PCR. And the purpose of this is to measure gene expression. So the first step is going to be RNA is extracted from the primary bone marrow derived macrophages, also known as BMDMs, or the peripheral brain endothelial cells. It's going to be called PBECs. And this is done using the nucleospin 96 RNA purification kit. After this, 400 nanograms of the isolated RNA was converted into complementary DNA. This is going to be called cDNA. And then you're going to be using the cloned AMV first strand cDNA synthesis kit. So this process involves oligo DT20 primers. And this is just done to bind the poly A tails of mRNA, which facilitates the reverse transcriptase process. After this, the specific primers for several target genes like CSF1, TNF, and AGR were designed. So each gene has a forward and a reverse primer listed, along with the expected size of the PCR product in the base pairs. And these are crucial for amplifying a specific gene of interest during the PCR process. So the RT-PCR was carried out using platinum, I think it's pronounced Cyber or SYBR green qPCR supermix. And the amplification process involved 40 cycles of temperature changes. Each change had a specific intent in mind. So the first round is going to be 2 minutes at 50 degrees Celsius. And this is done for the initial setup, so like a baseline. Your next 2 minutes is going to be at 95 degrees Celsius. This is done for denaturation. Your next one is going to be 15 seconds at 95 degrees Celsius, and this is specifically for the denaturation of the DNA. And then after that, you're going to be doing 1 minute at 60 degrees Celsius, and this is for the annealing and extension process. So the ABI Prism 7000 detection system was used to monitor this whole PCR process. So after this, we're going to be looking at the results of the QRT PCR that was normalized. So it was normalized to a reference gene called PIA, or peptidyl polydrol isomerase. And this is using the delta CT method. So essentially, this method just compares the expression levels of the target gene against the reference gene, and this is to account for variations in RNA quality and in its quantity as well.
All right, so for statistical analysis, just a brief rundown. So values will be shown by means of plus or negative SCM. Data analysis was performed with GraphPad Prism 5 software. For comparison of two groups, t-tests were used. Any groups that were used, that were more than two groups, one-way ANOVA and Tukey's multiple comparisons test, or two-way ANOVA followed by Benferrani post-hoc tests were used. For the SD, if infarctics, 25% of means, the chosen sample size of n equals 9 minus 11. Wait, frick. Okay, so sorry about that. So n equals 9-11 to detect 40% reduction of infarct size with a power of 0.5 and an error of 0.05 depending on specific experiments conditions.
All right, I know that was a lot of techniques, so we've gotten to the part that you've all been waiting for, which is the figures. We can finally put all of what you just heard about into application. And so starting off with figure 1, part A is going to be Ember.
I'm back. I know, you guys all miss me. So now we're going to get into our figures, and we're going to be really close to kind of wrapping up day 1 material. So be prepared, we still have day 2 afterwards. Alrighty, but so part A, we're just going to dive right into it. Be prepared, guys, we're going to get up close and personal because the camera is right next to the screen. So to start out, we have our y-axis, which is showing blood glucose levels, and that is showing in micromoles. And then we're going to go into our x-axis, which is looking at time after glucose injection. Okay, guys, I know I just called this one right here micromoles, but that's actually millimoles. So remember, M mmol. That's millimoles. When it has like the funky, that's going to be your micro. Anyways, we're going to go back into figure 1, and then as we're looking at this, we are looking at normal glycemic, which is going to be in these open-ended circles, and then hyperglycemic, which is these closed circles. And so figure A, this one is looking through just the impact of blood glucose levels set after a period of time. In this case, we're looking at the 0 minutes, so that's before our injection. We're going into 30-minute intervals, and then again after the 50-minute intervals. And then from here, as we can see, if we go in a line with that 30-minute one, obviously this line isn't covering it, just so you can still see which one's the normal glycemic and which one's the hyperglycemic. But right here, we have 3 asterisk marks. So the asterisks are representing what different values and figures that you'll see throughout the remainder figures have statistical value and importance in it. So when it's just the 1 asterisk, it's important, but there are areas where it relates higher and supports our hypothesis more or goes against it. So 3 is a very high setting of it. So as we can see, from the 0-minute marker, our blood glucose levels in both the normal glycemic and hyperglycemic were even. And then as we go into 30 minutes, there was a sharp increase in that hyperglycemic. As we can see, that one is right in line with the 15, whereas our normal glycemic is going to stay normal, as you can see right in here. And then from here, we have a decrease in both our normal glycemic and hyperglycemic. Our normal glycemic at the 50-minute mark, we can see it's only a slight decrease if we extend the line from where our point was. At the 30-minute mark, it's a slight decrease, which is why the statistical importance and value of this one isn't going to be as high as when we look at the hyperglycemic, because as we can look at that, if we draw our line from the 30-minute marker, there is a big difference that we can see right in here. And so again, that's just part A, showing the difference of blood glucose levels between the normal glycemic and the hyperglycemic. And then really quick, just to kind of sum it up, the technique that we used for figure 1, that is the measurement of the A-dicarbonyls. So just that we can kind of tie it into the techniques, and just that we can kind of put an interface. This is the technique that Brad had went over previously on in the presentation.
Now we're going to be discussing figure B. So in figure B, we're looking right here. Here is the infarct volume. The white right here is going to represent normal glycemic, while this dark one is going to represent hyperglycemic. As you can see, the results indicate that hyperglycemic significantly increased the infarct volumes in mice subjected to permanent distal middle cerebral artery occlusion, or the MCAO, compared to the normal glycemic mice as shown here. There is quite a difference between the two. This suggests that the elevated blood glucose levels negatively affects the extent of brain damage following ischemic stroke, and this reinforces the hypothesis this reinforces the idea that hyperglycemia exacerbates the consequences of stroke, which leads to larger areas of brain tissue damage.
All right, thank you, Ember and Makana. Moving into part C, first we're going to identify these axes here. So we've got on the y-axis, we've got this infarct area, which is talking about the size of brain tissue damage, and this is in square millimeters. And then moving on to the x-axis, we've got the distance from the anterior commissure, which is just a reference point that they used in the brain in order to decide where this infarct area was coming from. That's why it's got a peak point here. So this is like where the anterior commissure was. This is moving in one direction, and this is moving in the other direction right there. And so that's why it peaks like that. But as you can see, we're looking at this normal glycemic versus the hyperglycemic. I'm going to try to color code this here. And so as you can see, this hyperglycemic line is quite a ways above our normal glycemic line. Oops, sorry, it's still in green. I'll try to color code this here. All right, I know that's kind of a lot. But this is saying that the normal glycemic and the hyperglycemic, looking at the two, the hypoglycemic is consistently higher than the normal glycemic, meaning it's got more infarct area and more brain damage. And this is showing how these high blood glucose levels are going to have more of that brain damage following stroke, which we will discuss more once we get to our results. And starting us off is Ember.
All right, guys, so we're going to go ahead and just do a little recap of part A from figure 1 and get into our results of this figure. Let's get a little more centered. There we are. Alrighty. So again, part A, it overall was just showing that the hyperglycemic mice did have a higher increase in that blood glucose level, and that was seen over the time period of the 0 minutes, which is when it was first injected, going into that 30 minutes where it had that really sharp increase. And then when it went to that 15-minute time period where they took that result, it did drop in numbers, which it should have, because that's showing that the body and that inflammatory process is doing its job in kind of activation there. But no matter what, it did see that the hyperglycemic mice did have a dramatic increase when compared to those normal glycemic mice. Now we have Makana with part B.
Hello. As stated earlier, Part B did show that elevated blood glucose levels did negatively affect the extent of brain damage following ischemic stroke, and that hyperglycemia does exacerbate the consequences of stroke, causing larger areas of the brain to be damaged due to the hyperglycemia.
All right, and again with part C, as we saw, there was a hyperglycemic consistent arc that was higher than the normal glycemic mark for the distance from the anterior commissure, which was that bundle of nerve fibers that they used as a reference point in the brain because of the neuron activity there. And this suggests that the high blood glucose levels cause a greater brain damage following stroke in these mice. And
that concludes day 1.
Alright, hello everyone. So this is going to be our day 2 slides. So let's begin with a day 1 recap. So we explained a little bit of like what hyperglycemia is and its causes, and how hyperglycemia is increase the infarct volume and decrease the number of protective non-inflammatory monocytes and macrophages in the ischemic brain. And basically we just talked about like, you know, what ischemic brain is and like ischemic stroke and all that stuff that we're going to be researching more in this presentation. How agar interfered with polarization of macrophages to a non-inflammatory phenotype. And we also discussed a bit about figure 1 and the results. And to reiterate the results, so basically hyperglycemic mice had a higher increase in blood glucose in an hour compared to normal glycemic mice. And another thing is hyperglycemia appears to lead to greater stroke-induced brain damage in mice because the larger infarct area in the mice suggests high blood glucose worsens outcomes in ischemic stroke. So now we're going to be looking at figure 2, graphs A through F. And this is from the results of the flow cytometric, which is basically the characterization of immune cells in the ischemic brain under hyper and normoglycemic conditions. So as we can see here, graphs A through F are results from blood samples that were taken 48 hours after MCAO, which is basically middle cerebral artery occlusion, which creates stroke model in animals. And just a rundown so that we can like understand what each of these things that I'm circling here, what they mean. So CD45 is PE labeled rat anti-mouse, and LY-6C is APC labeled rat anti-mouse. CD11B is per-CP labeled rat anti-mouse, and LY6G is labeled rat anti-mouse. And basically what we're seeing here, so I'm going to be circling these just so that we know what each of them are talking about. I'm going to go into a little bit more depth as like we go along when we break these apart. But basically, as you can see here with all these like colored dots, so the warmer the color is the higher density of cells as a result of these expressions of the gated cells as shown in the graphs, comparing them side by side. There's a polarizing results when it comes to these cells under normal glycemic and hyperglycemic expressions. So like I said, we'll be breaking this apart so then we can get like a better understanding of this. So right here, I explained before. So for parts A and B of figure 2, we have CD11B and CD45, and we're comparing the two with when it comes to normal glycemic and hyperglycemic conditions. So as we can see here, in these areas, we can see that there's a significant change, like the cells increase in basically in density, which means they change in density as a result of the expression of these cell expressions right here. And then we compare this to hyperglycemic, which we can see here. There's more density. Like I said, the warmer colors represent like more density in the cells. So as we can see here, as a result from hyperglycemia, we can see that it's a lot more. There's a lot more cells that like express more density because of these cells expressions. So now we're looking at parts C through F, and as we can see here, this is comparing LY-6G and LY-6C expressions, and as we can see here, there is less of a cell density change in these graphs compared to A through B in the previous slide, but we can still see that there's still a significant change. And and just like graphs A and B, we can see that there is a lot more cell density in the hyperglycemic labeled graphs, which is D and F, compared to C and E, which is a normal glycemic conditions. So now we're moving on from graphs A through F. So now we're moving on to parts G through J. So basically, G through J is a quantification of the ones that you can see on screen, where microgylia, non-inflammatory monocytes and macrophages, inflammatory monocytes and macrophages, and neutrophils. And basically, just to briefly talk about each one. So microgylia is basically immune cells. Non-inflammatory monocytes and macrophages are first line defense. Inflammatory monocytes and macrophages are cause inflammation and basically not for first line defense. So and neutrophils are basically white blood cells. And we can see here, based on the key right here, so white is normal glycemic and hyperglycemic. As we can see, so basically H has the most significance, as we can see here from the asterisk, I think that's how you pronounce it. So so we can see here that when it comes to hyperglycemic conditions, when it comes to non-inflammatory monocytes and macrophages, it's less expressive than as we compare to normal glycemic, and this could be the case for H through J, except for G for microgylia, which could be because of the fact that they're immune cells. So that might be a reason why there's a change. But for H through J, we can see that hyperglycemia basically has lower or causes these three right here, H through J, had to have lower expression. Okay, just to summarize the results that we just analyzed from the graphs. So basically, the percentage of CD45 low and CD11B cells in the extreme hemisphere that were mostly LY-6C low were not significantly affected by hyperglycemia. as we can see in the previous graphs, and CD45 high and CD11B cells were detected only after MCAO in the ischemic brain and represent bone marrow derived cells. And among CD45 high, CD11B plus cells, inflammatory monocytes and macrophages and neutrophils did not significantly change in number under hyperglycemic conditions. Again, as you can see from the previous graphs, and hyperglycemia reduced non-inflammatory monocytes and macrophages in the ischemic hemisphere, although the infarct volume increased under the same conditions.
So in this figure here, we're going to be looking at ABC out of 1100. I don't know, there was a ton of these ones. But you can see here, this green fluorescence down here is going to be showing the IBA1 positive genes, and you can see all these little bitty arrows are pointing, like all these little specks through here are macrophages that have the positive genetics for that IBA1. And in this red one here, we're going to be looking at the ARG1, which is another, combination of genes, but it's the same thing with the green. It's what we're looking for. And then over here in this one here with the orangish greenish kind of color, it's actually just an overlay of both of these ones. So if we add these, it equals this one, and this is the one marking both the ARG1 and IBA1. It's just double positive for the anti-inflammatory macrophage expression. So now we're actually going to get into the good part of this, is what we're actually looking for. So that last one was just that normal glycemic. This time we're going to be looking at the hyperglycemic, and you can see there's already a reduction in all of these that, it's the same thing. This is going to be that IBA1, this is going to be that ARG1, and this is going to be the merged, where they're all marking that fluorescence for the positive macrophages. You can see there's been a reduction, which just shows there's less of those expressed. So for part G and H of figure 3, we're going to be looking at these two graphs here, which is going to be the combination of that ARG1 and IBA1, which is just that right side of the last two slides, and the total number of just the IBA1 cells. So over here we can see there's a big difference, right, in these lines on the histograms, but what that is showing is just that there's going to be an increased number, or a decreased number, sorry, of those anti-inflammatory macrophages in the hyperglycemic side here. So with this combination of the ARG1 and the IBA1, it's not actually going to be affecting the number of macrophages, but the number of macrophages that differentiate into the anti-inflammatory ones that we're looking for with these strokes. That's just going to lower that effect, lower the infarction, and it's actually, I want to say, these are measuring the amount of those macrophages in the area surrounding the actual infarction. And then this one over here, there's not any significant difference, again, between when this is just the IBA1, this is without the ARG1, so there's not going to be a huge difference, but when these two combine, that's when we start to see that difference, that lowering, that lessening. But over here, there's no statistical difference. They're exactly the same. It's no different. So the overall results from figure 3 just show that hyperglycemia actually does reduce the number of anti-inflammatory cells in the area around the infarction up to 48 hours after the stroke without affecting the actual number of macrophages. So that's what I was just showing you in that previous histogram. And then the total number of the IBA1 plus cells didn't change, which is just another, the number of the cells didn't change, but the number of ones that are expressing the anti-inflammatory phenotype did change. It actually decreased, which is what we were looking for.
All right, so moving on to figure 4, this is going to be outlining the experimental process that I kind of talked about earlier with both the mice and the stroke model in the techniques. So starting out in step 1, we've got this 10 GY radiation, and the purpose of this is to suppress the immune system of the mice. They're wanting to completely isolate whatever cells they're looking at, and so this is going to target that bone marrow that is producing the immune cells, and this happens in a 24-hour, or it incubates for 24 hours, and so that we can move on to step 2. And step 2 is going to be that bone marrow transplant. I'm just going to write a little acronym right there because there's not really a label for this one, but that's what the step is outlining. So we've got this right here, which is going to be our green fluorescent protein labeled monocytes, which the GFP is going to make those visible to be able to monitor those levels, which I'll actually show more with in the next slide when we talk about the immunofluorescent microscopy. And so this allows the researchers to notice these cells once they are in the mouse's system. And this has to happen over a 6-week period in order for the bone marrow cells to establish and for the monocytes and macrophages to be replaced from the original cells. And then this is this final step here that is significant to all of the tests that will come later and all the ones that actually have been a little previously discussed. And so this is the middle cerebral artery occlusion, and it actually gives a little bit of a visual right here of where exactly they're going into the mouse in order to get access to that artery and to induce the stroke. It is a very invasive process. I feel like this picture makes it look a little less complicated than it is, but hopefully this helped clear up anything that people were confused about with this experimental design. And so it's laid out in these steps for you guys. All right, so now we've got parts B through I. I don't know why it's broken up into so many little squares, but I will break it down for you, don't you worry. This is the immunofluorescent microscopy, and it looks a little crazy, and we don't know what's going on at first, but on here, this little like y-axis, pretty much, I mean, it's not a graph exactly, but here it's getting broken into the normal glycemic versus the hyperglycemic, which is important for us to be able to recognize the differences of. And so we've got this GFP, which is the label for the monocytes and the macrophages, which I talked a little bit about in that second step for the bone marrow transplantation in the last slide. And so these little arrows, the little like yellow and white arrows here, they are pointing to specific cells that are expressing this GFP in the immune cells in the brain tissue. And so this is showing us where the monocytes and the macrophages are existing. And between the hyperglycemic and the normal glycemic, it's not very significant difference. But then we move on to this arginase 1 that I think Ember talked about a little bit earlier. And it's associated with the anti-inflammatory response, that M2 type macrophage that we've been talking about. And this phenotype is what we want. We want the non-inflammatory type to then protect the brain after the cell damage. And so as we can see, this normal glycemic has a lot more positive results of this marker than the hyperglycemic, so my little circles here, I don't know how many of a difference that is, but we've got more in the normal glycemic. And moving on to our little other marker here. So this stands for immune nitric oxide synthase. And if you know anything about this, this is one of the vasodilatory processes in the immune system that works with inflammation. So this is our pro-inflammatory response. So the difference here, we've got the anti-inflammatory response with arginase versus the inflammatory response with this INOS here. And these are the M1 type macrophages in comparison to the M2 type macrophages. And these are similar in both conditions pretty much, as you can see with the little arrows again. I won't mark all of them, but you can see they're pretty similar. And then if we merge them all together in this last slide, along with, they added an extra nuclear blue stain in order to be able to recognize the spatial relationship between the different cell markers and nuclei. So the biggest takeaway here is that there's more of the positive arginase 1 from right here in the normal glycemic versus the hyperglycemic. And that's seen more in this merged one, but I feel like it's easier to see in the little second column because there's not as much staining there, but that is that main takeaway there.
Alrighty guys, so now we're just going to go a little bit more into figure 4. This is going to be parts J through M, and this is kind of just putting into a histogram of what all the results that Mayah just explained went over. So if you remember in her step 2 of part A, that was showing GFP, and like I said in the immunofluorescence microscopy slide from day 1, it was going to come back. So just to kind of remind you guys, that stands for green fluorescent protein. And when we go right into this slide, that GFP, it's going to be the acronym in this first part. So we have our green fluorescent protein going into arginase 1. We have our immune nitric oxide synthase, and then cells just because that's what they are. And when you're looking at, oh, there we go. Alrighty, so when you are looking at these different histograms, so between J, K, L, and M, what's really important to note is where the positive and negative signs are on each of the y-axes. So if we look at J, we have a positive on GFP, negative on arginase 1, and then negative on that INOS. So that's really important to note. So we'll just write right here, negative, positive, negative. When we go into figure K, we're seeing it as a positive, negative, positive. So again, positive, negative, positive. For figure L, we have a triple positive. I don't think we need to write that one down just because we can kind of see what that one's showing right there, and with it all being positive, it's pretty easy to remember that. And then in figure M, in figure 4 part M, we have it as a positive, positive, negative with those different protein types right there. And so what we're really looking at in these figures is the distance between our normal glycemic and our hyperglycemic. So if you look in this bottom left section right here, I just circled it in green. So that is our little key to see which one's representing normal glycemic, which is our white boxes. And then you can see that our hyperglycemic are these black shaded in boxes. And when you look at figure J, there is not a statistical significance within the difference between these two forms. So when we have that GFP protein is present, which all of these are showing it as present because that's the staining that was used in it. But so when we, sorry, I messed up on this J one, I just realized. This is actually a positive, negative, negative, not this one right here. So sorry about any confusion here. I'll go ahead and erase that so we can kind of see it a little better. Alrighty. So anyways, with that, we can just kind of see when arginase 1 and the INOS are not really represented, that there is no difference. When we go into figure K, we can see and start to see that difference in there, and that's the statistical difference that we're seeing right here. As you can see, our normal glycemic is at the 50 marker, whereas the hyperglycemic is roughly around about 25. In L, this is where we're going to see our biggest difference, and this is when all of the cell bodies are present in that one. So with here, as we can see, we do have the highest jump in here. So our significance factor is going to come into play a lot. And then with figure M, we still see that significance level even with the INOS being at a negative in that arginase 1. So overall, this is just really showing the impact that arginase 1 has within these different normal glycemic and hyperglycemic conditions. And we're just kind of looking at the peri-infarct tissue, and that's going to be just kind of like an overall resemblance of what's going on here. And then now we move into parts N through Q. And this is going to be the final part for figure 4. So when we're looking at this, it's really important that we understand figure N. Now, this is going to be where we start looking into those cerebral cryosections that we mentioned in those techniques. And it's really important because we are grouping this into 3 sections. So we have our motor cortex, which is going to be this top area, our striatum, which is the middle, and then our insular cortex. And then what they did is when breaking this down to look into the results, they just went ahead and took each section and made their own little graph for it. So as you can see, O is the motor cortex, P is the striatum, and Q is the insular cortex. And what they saw through here was that in the motor cortex, that's when we had our greatest increase between the normal glycemic and hyperglycemic conditions. And again, because arginase 1, as shown in the last set of figures, had the most significant impact, now we're just going to look in at the GFP and arginase. And so that's what they're really focusing in on right now. And figure O, or part O, there we go. So that one is just looking in it, and we see that it did have the greatest increase, which is why it has the 3 asterisks on it, and that one's just showing that it is of most significance. And then even though in figure P it has an increase between the 2, it's not that great. So yes, it is showing that the striatum does change and that the normal glycemic conditions are a lot better for controlling the brain and then the inflammatory process in there. It's not as much as what we can see in the motor cortex, which is going to be the main part looked into. And then finally, when we look in the insular cortex, it's actually a slight increase in the hyperglycemic conditions, but they do relatively stay the same, which is why that one we do not see it as prevalent there. And then we're going to go ahead and move on into our results for figure 4.
All right, I'm coming back to you guys. All right, so when it comes to the first part of this figure, since it was just the experimental design, there's not really a lot of results there. I also did forget to mention earlier that between the bone marrow transplant and the MCAO stage, that is where the injection of glucose is going to happen to differentiate that hyperglycemic versus normal glycemic group of mice. Also, the INOS does stand for inducible nitric oxide synthase. I think I said immune earlier. That's just the way that I had remembered it, so I apologize for that discrepancy. And anyway, so going into the part of the immunofluorescent microscopy results. So again, this was showing the monocyte and macrophage markers in the brain from the normal glycemic and the hyperglycemic as seen on those different panels in the box kind of graph-like figure that we saw. To represent the different markers and their different combinations. So this immunofluorescent technique supports the hypothesis of these researchers by suggesting that hyperglycemia impairs the polarization of the macrophages toward the M2 anti-inflammatory pathway, which is the pathway that we want. Then the phenotype is also in that stroke-affected brain tissue, which is what we're trying to figure out to protect the brain more than damage it. And again, it's talking about this reaching that threshold with the inflammatory process coming from helping the cell by cleaning it up versus then damaging the cell by causing too much vascular permeation that is messing up things within the cell. And yeah, so that's a less favorable immune environment overall.
Alrighty, and then just to kind of go over the remaining results, so what we saw was that those non-inflammatory macrophages of the arginase 1 positive and then the inducible nitric oxide synthase, which I also said immune, so sorry for any confusion on that one. But we did see that those had a reduced in density there, which is what those histographs are showing. So the cells are measured in millimeters, and so that's kind of what it was showing there and how the size of the inflammation in those normal glycemic conditions had decreased, and then why it went up so high, it's just that was showing how much it decreased. That wasn't necessarily showing the size of it, but the decrease factor in the density. And then the peri-infarct tissue, that was us looking into the motor cortex, striatum, and insular cortex. and that kind of just showed how they were all being analyzed separately and broken down into it. Just kind of to recap on that one, we did see a significant decrease in the organized 1 cells in the motor cortex, and that was the one that had the increase in significance in there. The other ones, yes, there was a slight increase, but it was not the dominant factor. And then finally, we just wanted to make sure that we really enhanced that there was no difference in that insular cortex, and that slight increase in the hyperglycemic conditions, it was just something that comes with the results, and that it was just a part of the testing that they had found. And yeah.
All right, so figure 5 here, parts A, B, C, and D, we're going to be looking at the wild type controls, which is just a regular old mouse, and then that CD11B, which is a mouse that has been genetically modified with this diphtheria toxin receptor, which, looking at all these panels down here, the diphtheria toxin is being used on the B and D panels, and the salt water is just going to be used on that A and C. But we can see when this diphtheria toxin comes in contact with the diphtheria toxin receptors, it's actually going to cause those monocytes to get rid of whatever is there, it's bothering it, which is just exactly what would happen in an ischemic stroke, is what we're hoping to happen. But you can see here this LY6C, in this one it's increased, but it's significantly more than over here. So this LY6C high gene specifically is the one that we're looking at, because that is the one that is going to inhibit the, actions of the monocytes in those wild type mice. And that DTR group, that salt water is just a control for the DTR group, and then it's reduction in that expression in the LY6C gene, which just effectively depletes the monocytes, and they're doing their job, providing direct evidence that monocytes, especially of this subset right here, impact the hyperglycemic infarction size. But you can see here in the LY6C high gene on just the salt water is more, but with that diphtheria toxin, it's even expressed less. So we're going to see less of an impact in this one than was in here. And we're really only looking at this diphtheria toxin because that salt water in both of these is just a control to see what would happen if there was no diphtheria toxin. And there's no receptors here, so it shouldn't be expressing as much. But with this genetically modified receptor on those cells in the mice, we're going to be seeing a decrease in that expression. Okay, so for figure 5, part E, F, G, and H, we're going to be looking at the monocyte expression and the neutrophil percentage. So looking at this graph here, looking at the LYC6 low cells, there's an 87% reduction between the wild type and the CD11B DTR genes, or monocytes, my apologies, in the mice treated with that DT. So this positive down here is showing what I was just drawing on that last slide, where the expression of the DT is actually going to drop significantly right here, 87%. This is just the one that was treated. This is the one that was treated with salt water. This is the one treated with that diphtheria toxin. And then looking over here at the LY6C high, that one I highlighted last time, there's actually an 83% reduction between between the non-diphtheria and the diphtheria toxin down here. You can see there was a bit of a difference that I had brought up last time, where the diphtheria toxin it expressed some more on those monocytes, but it's a big drop off, which is what we were looking for in the experiment. And then graph G here is comparing the neutrophil percentage in the wild type and the DTR mice. So you can see it's about the same. There's not a big difference. I mean, I can draw you a line there. It's about that much statistically. It's not a huge difference. It's pretty insignificant. They didn't really even talk about it in the presentation, but so the graph H here, this one, is comparing the infarction volume between the CD11 and the wild type mice, and you can see that there's no statistical difference between the normal glycemic and the hyperglycemic in that section, which is exactly what we were looking for. It's important because right here you can see in the wild type that infarction volume in the hyperglycemic is going to be more. It's going to be larger. It's going to be more damaging. I mean, it's just going to be a worse stroke. There's going to be more damage to the brain and the function of the brain. But over here, there's not any statistical difference. That's what this NS means right here, where we're looking good, looking up. So now looking at those figure 5 results, we're actually going to see that the DT treatment in the CD11 DTR mice significantly reduces both the LY6 low and the LY6C high monocytes by over 80% each. We're looking at 87 for the low and 83 for the high. I mean, they're both significant. That's what we're looking for. The neutrophil populations, there was no changes in the neutrophil percentages between monocyte depletion in both of them. They were exactly the same. That was the graph I showed you before. And then the infarct volume right here, we're looking at the hyperglycemia significantly increased that infarction size in the wild type mice, but in the DTR mice, there was no significant difference in the infarction size between the normal glycemic and the hyperglycemic mice. This just shows that the monocytes were able to prevent the worsening of the stroke even in the hyperglycemic through that diphtherotoxin receptor that is on the monocyte.
All right, everyone, so now we're going to go ahead and get into figure 6. So here we're going to be looking at parts A, B, and C, and the technique that was used through this one was the measuring of A dicarbonyls, which if you remember, that was kind of addressed with Brad. So in here, we are looking at, on our y-axis, we have our glyoxyl in figure A, we have methylglyoxyl in figure B, and then we have our 3DG in figure C. And now on the y-axis, these are all going to be measured in nanomoles, and it's going to be nanomolar to the -1. Now, as we can see here, when looking at them, we are seeing our normal glycemic is in our open circles, and our hyperglycemic is in these closed colored in ones. And what we're really looking at is just seeing how the different normal glycemic and hyperglycemic infusions work with the different glyoxyl, methylglyoxyl, or 3DG over time. And so we tested it at the 0-minute intersection, which is our first point for all of them. So that's right on that y-axis. And so that is before we had injected them with that hyperglycemic in it. And then we're going to go into our 30-minute time zone for all of them. And then we'll check again in at the 50 minutes. And the most important part to note from this figure is in the 3DG, our 30-minute and 50-minute markers are the ones of most significant. I know it's hard to see in here because of how small the circles are, but if you look closely, you can see this bottom line. I'll go ahead and kind of box it right here. In our 30 to 50 minute, that is our normal glycemic conditions. And then if you look above that, it's our hyperglycemic. And that hyperglycemic is where we did see the big surge and the most sufficient change in there. And then we're going to head into parts D, E, F, and G. And now these are kind of just putting into another perspective of what we just went over in the last few parts. As we can see here, we'll go ahead and we're just going to jump to figure F really quick, or part F. And again, we have these brain cryosections, and those ones, again, we are looking at the motor cortex, striatum, and insular. And so that's what the 3 parts are. If you really zoom into it, you can see the different sections of them right there. You can kind of see where the color changes, and that shows the different sections. When we are looking at the figures, again, we are looking for those asterisks. So I'm going to go ahead and actually erase what I just drew for our cortexes so we can kind of see those asterisks and what they're really showing. So as you can see, when we start at figure D, the hyperglycemic, which is the ones that are fully shaded, had a great increase compared to the normal glycemic at the 50-minute time frame, and that was with the glyoxyl, and that one we were measuring the nanomoles per milligram of protein. So that is what this right here is showing on our y-axis. And then E did not have any significant showings in it, and then the study didn't go too much into it in the article and what they found, just because they found that because they had no change in the methylglyoxal, that it wasn't important enough to kind of really focus and center it on. So we're going to go ahead and move into F, and here we see that we're looking at the wild type and then the agar, and it's really important to note that the agar, it's at that negative stage for both part F and G. And then again, we are just showing here that the hyperglycemic conditions in part F are higher than the normal glycemic, so we can see that here and here. Although the one in agar has a slight increase, it's still important to note, and that did show significance, just because it's showing the impact agar has on it, which is what our study was focusing on. And then with figure 6 part G, we are just seeing that the wild type, which is the mice that were not injected under those different conditions and stuff that didn't go through the agar processing, it is still seeing that our normal glycemic had that increase into them.
All right, thank you, Ember. Moving on to our part H through M. So we've got a lot of graphs here, but I'm going to break it down into our anti-inflammatory processes right here, and then our normal inflammatory right here. I'm just going to do inflam. There you go. Hopefully that's helpful. But so we've got our BMDM, which is those bone marrow derived macrophages, and we're looking at the wild type versus the agar knockout. So the agar knockout is going to be without that gene, where the wild type is going to be with it. And for our anti-inflammatory processes, we've got all of these anti-inflammatory markers, and under a treatment with interleukin-4, which is on all of these, and it is going to be that anti-inflammatory process with those M2 macrophages being polarized. And so as you can see here, we've got some statistical significance for the agar knockout being higher than the wild type. And then at the bottom with the inflammatory process, we've got this LPS treatment, which stands for the lipopolysaccharide, and this is promoting the M1 polarization in the macrophages, which is that inflammatory response. So again, we want this anti-inflammatory response in order to get the protection, that neuroprotection that I was talking about earlier, and we don't want this inflammatory response. So as you can see here, there's no statistical significance with this TNF gene. That stands for tumor necrosis factor. They don't really go into why that wasn't significant. I don't think they really knew. That might be a good thing to look at in future research. But as you can see, we've got our wild type is higher in the inflammatory process, where it was lower in the anti-inflammatory process. Therefore, we want the knockout agar, meaning we don't want the gene, if that makes sense. Sorry, it's a little bit of a double negative. And then this final LPS versus interleukin-4 testing is showing that the wild type is more common in the inflammatory response, again supporting that fact that we do not want that agar gene. Okay, and just reiterating, these two, so this TNF and this other interleukin, are the inflammatory markers, where this is looking specifically at the agar mRNA for just the wild type. So we've got kind of like this inflammatory response right here, this anti-inflammatory response right here, and then we're just looking at the wild type for this one.
And then just to kind of reiterate the main points of figure 6 and go over the results, what we really saw in here was that agar increases the infarct volume in hyperglycemic stroke, and that one is just showing that there was that increase, which is what we saw with those asterisk markings. And then we also saw within this that the 3DG, which was the third data tip that we had shown, or the graph, and that was just showing that there's a significant increase in the hyperglycemic conditions, And that's mainly due to just the fast metabolism of MG. And that's what the researchers had said and like deemed it to be as. And then our hyperglycemic agar mice, they had a smaller infarct than the wild-type mice within the 48 hours of the MCAO. And again, that was just looking into the motor cortex, the striatum, and the insular cortex. And along with that, it kind of plays into figure G, where it was showing the agar knockout gene, and that one was just showing that there wasn't significant change, meaning that the hyperglycemia did not reduce the non-inflammatory of the arginase 1 and IBA1 macrophages in those peri-infarct tissues.
All right, and referring to the last little array of histograms, the expression of the agar may determine the macrophage polarization as seen with the more mRNA in the wild type with the inflammatory pathway versus the anti-inflammatory pathway. So overall, these findings suggest that the absence of the agar gene promotes an anti-inflammatory response, reducing the pro-inflammatory response, which is what we want in the bone marrow-derived macrophages, therefore being more protective for the brain following the ischemic stroke.
Okay, we're going to be reviewing figure 7, part A, B, and C. If you're looking right here, it shows the MG treatment increased TNF expression in peripheral blood endothelial sites, or PBECs, under controlled conditions and after oxygen glucose deprivation. You can also see this right here in figure B, where MG reduced mRNA levels in colony stimulating factor, or CSF1, and the release of CSF1 from PBECs here. You can see that the control in the OGD is less here and here, yet increased here and here. Okay, we're now going to be looking at figure D right here. So figure D is investigating the crosstalk of PBEC with BMDM in secretion by CSF1 right here. And so it's being treated with the conditioned medium. As you can see, it's coming over. The conditioned medium of PBECs that were stimulated with MG and OGD. Okay, moving on to figure E and F. This one has a lot going on. So right here, we're looking at our relative retinal mRNA. These and the relative CHIL3 mRNA, they are both inflammatory markers. Up here, we have our BMD, and these right here is going to be our PBECs and BMDM. So looking at this, the BMDM, the expression of retinal and CHIL3, which are the typical marker genes of inflammatory macrophages, it was diminished by the CM and MG, and which is oxygen glucose deprivation for OGD, and challenged PBECs. So a comparable effect was obtained by adding CSF, one neutralizing antibodies to CM, which is the controlled medium, and expression was normalized to controls that did not receive the CM. So here you can see the negatives and the positives, which represents each bar. Okay, now we're going to be looking over the results of figure 7, A, B, C, and D. So for figure 7A, treatment of primary brain endothelial cells, or the PBECs, with MG increased the expression of the pro-inflammatory cytokine TNF and decreased the expression of colony stimulating factor 1, or CSF1, which suggests that the MG skews macrophage polarization towards an inflammatory state. For figure 7B, it shows that under normal and oxygen glucose deprivation, which is that OGD condition, MG treatment reduced CSF1 secretion from PBECs, indicating a potential mechanism by which MG impairs non-inflammatory polarization of macrophages. For figure C, it's going to be similar to the mRNA expression results. CSF1 secretion was lower in PBECs treated with MG, further supporting the idea that MG negatively affects CSF1 production. And for figure D, BMDMs, which is the bone marrow deprived macrophages, are stimulated with conditioned medium from PBECs treated with OGD and showed increased expression of non-inflammatory markers, retinal and the CHIL3. So this indicates a shift towards a non-inflammatory phenotype. For figure 7, E, and F, the results for E was when BMDM was treated with conditioned medium from PBECs that were co-treated with MG and OGD. This shift towards a non-inflammatory phenotype was prevented, which demonstrated the negative impact of MG. And for figure 7F, the reduction in retinal and CHIL3 expression was also observed when CSF1 neutralizing antibodies were added to the conditioned medium. So this just reinforced the presence of CSF1 being crucial for promoting non-inflammatory polarization. So overall, the figures illustrated that hyperglycemia through the action of MG interferes with the non-inflammatory polarization of macrophages by reducing CSF1 expression in endothelial cells, which just ultimately contributes to pro-inflammatory environments in the ischemic brain. Next, we're going to be looking over figure 8. So in the event of cerebral ischemia, inflammatory monocytes and macrophages enter the brain tissue, as seen right here, and undergo polarization into a protective phenotype, which is this guy right here. And this protective phenotype is crucial for healing and reducing damage. Next, we're looking at hyperglycemia, which is right here, that increased glucose. Hyperglycemia disrupts the normal process, impairing the ability of macrophages to polarize into the protective phenotype, as they did here. Next, we're going to be discussing the role of agar. So this guy over here, so the advanced glycan end product receptor, or agar, negatively influences the polarization process because it inhibits the conversion of macrophages to the protective phenotype, and this interferes with the beneficial effects, which worsens the condition of someone going through ischemic stroke. So next, MG, which is a harmful byproduct of glucose metabolism, further complicates this matter by decreasing the expression, and it releases decreasing the expression and release of cytokine CSF1 from the endothelial cells. So if you're looking right here, here's our agar. And so the CSF1 is important because it encourages macrophages to polarize into their non-inflammatory and protective state. So if it's inhibiting the CSF1, it's reducing the non-inflammatory state, which further damages. So overall, the passage just describes the cycle which hyperglycemia and the associated metabolites disrupt the protective polarization of immune cells in the brain, which has the potential to worsen the outcomes of ischemic injury. Okay, and looking here at the results, since we were using a illustration, pretty much everything I said in these results was demonstrated over the inflammation diagram, but essentially the main points is that the inflammatory macrophages enter the brain and they should be protective, but the hyperglycemia hinders that polarization in addition to the agar. And the MG, which is a byproduct of that glucose metabolism, will also reduce the expression of the CSF1s, which further causes damage.
So revisiting our hypothesis, which our initial hypothesis is basically, hyperglycemia would downregulate the effects of macrophages and monocytes. And looking back, the data suggests that hyperglycemia exerts detrimental effect through monocytes and in that monocytes infiltrates the ischemic brain tissue and polarize under normal glycemic conditions to non-inflammatory macrophages and have neutral protective properties. So in the end, monocytes and macrophages are essential parts of infiltrating the ischemic brain tissue, and hyperglycemia has a deep effect on that. And just to reiterate, just to like clearly state the hypothesis, so basically our hypothesis was supported.
So overall conclusions for the paper, it just shows important insights into how hyperglycemia makes stroke damage worse. The researchers actually found that when blood sugar was high, it changes how the immune cells called monocytes or macrophages work in the brain after a stroke. And these cells normally help protect the brain, but high sugar levels interfere with their ability to do that, and it actually makes it worse in the long run. The study discovered two key players in this process. Those harmful metabolites that I spoke about earlier, the beta dicarbonyls or the agar, which are just the receptor on the macrophages. But when the researchers removed the agar from the mice, the negative effects of that high blood pressure were reduced, which means that targeting these specific molecules could potentially help stroke patients in the future that have that high blood sugar hyperglycemia. The findings are significant because current treatments like insulin haven't been able to successfully solve this problem, and by understanding how exactly hyperglycemia damages the brain during a stroke, researchers might actually be able to figure out new treatments that could help us fight that better and help save lives in the future.
All right, welcome to my final slide of the presentation, the limitations and suggestions. So we've kind of been going over these as we've gone throughout the presentation, and the biggest limitation is that this exact model could not be done on human subjects. As explained before, it's a pretty invasive process in order to stimulate that stroke in the mice, and we're not going to go decapitating humans or drilling holes into their heads. So in order to be able to look at this more in human models and look at their specific biological responses, it would be suggested to do clinical trials and less invasive means in order to look at this. And this study also focused mainly on the agar mechanism, and there's other glucose-related metabolic pathways that we could look at, and so future experiments could look at that more in order to completely understand how this hyperglycemia is inducing the stroke, or not inducing the stroke, but influencing the stroke. And as well as it focused mainly on acute hyperglycemia. I know a couple people brought up the relation to diabetes, but because this was an induced acute hyperglycemia in the mice, it wasn't able to try to influence those more chronic conditions as seen in humans, and diabetes is becoming a more significant role in the healthcare field, and so that would be something that we would like to look at more, as well as differences between gender and age. As seen in humans, it's seen that women are more likely to have strokes, but men are more likely to have them at a younger age, and so it would be interesting to try to control these different genders and ages to determine like what is causing it in different ages or different genders in order to really pinpoint those conditions and to help people more depending on their specific circumstances. And finally, environmental and lifestyle factors. We were not able to look at this with the mice specifically because, we're inducing the stroke in them, and there's not a way to see how their diet and their exercise and stress and that kind of stuff. And so if we were wanting to look at this more with humans, it would be a good suggestion to look at those lifestyle factors as well.
From the Christian worldview perspective, we chose Proverbs 4:7, which says, "The beginning of wisdom is this, get wisdom, though it cost all you have, get understanding." We chose this first because we felt that it was a reflection that scientific research is a tool to serve your neighbors. The more wisdom that you acquire, the more you can apply it to science and better benefit the lives of others.
Awesome. Now we're just going to go into a couple frequently asked questions because you guys aren't able to ask us the questions, so we just came up with ones that had recently occurred. So our first one is, the research shows hyperglycemia being a treatment to ischemic stroke. How is this proved? And the hyperglycemia is used as a treatment because of the inflammatory monocytes being infiltrated into the brain tissue and polarizing during those normal glycemic conditions. So hyperglycemia is actually interfering with this, and it's lowering the inflammation of the arginase 1 or LYC6 macrophages. And so those are the ones that have the neuroprotective properties. So hyperglycemia is not necessarily as the full treatment to it, but as in a way, a helper for when it comes into that. And then our second question is, what do the neuroprotective properties mean regarding that arginase 1 and the LYC6 or LY6C? And so those, along with other non-inflammatory macrophages and monocytes, which is what the LY60 is, if you don't remember that from earlier on, but these are just able to promote that tissue repair and regeneration. And when these are depleted or damaged, it can increase the risk of hemorrhaging after ischemic strokes. So this is why we really want to increase those, so that way we can protect this and make sure that the ischemic stroke rates are actually lowered. And then, was there a specific technique that seemed to be the most influential? So one of the techniques that was used in a lot of the different figures and did seem to show the most results and influentiality was our measuring A dicarbonyls. So again, this is the one that Brad had mentioned earlier on, and this technique was just the most influential because research on how hyperglycemia impacts individuals with ischemic stroke is limited, and this technique kind of helps break down the non-inflammatory macrophages a little bit more in seeing how exactly they're impacted and over what course of time they are most prevalent. All right, everyone, and then just to kind of conclude, we have a Kahoot for you guys to do. We can't really pull it up right now because if we do, it'll end our recording session, but it is there, so you can go ahead and test your knowledge on our presentation and kind of test for your understanding. But that concludes our presentation. So thank you guys for watching, and we hope you enjoyed what you learned.
Bye!
Bye!
And here are the references.
Down below!
Down below! Check them out. Pretty cool stuff.